Jake, welcome to our flagship studios. We're delighted to have you here this morning to tell us a little bit more about Quotient Therapeutics, a company that Flagship announced late last year and is rather unique in the Flagship ecosystem, both in terms of its science, but also geographical footprint and formation. And also, as I understand it, the second company that you have co-founded in the field of genetics. So to get us started, tell us a little bit about somatic genomics. What does that mean? I have heard you say before that we have long assumed that we have one genome, but it turns out that we were off by a trillion-fold. How could that be?
Well, thanks for the invitation, Lindy. It's exciting to talk about Quotient. So this blew our minds when we realized that this was the case. Growing up, when we learned about biology, we all learned we have a genome, and that genome codes for everything that goes on inside of our cells in our body. But what the textbooks didn't realize, and what has recently emerged, is that it turns out each cell in our body has its own genome. So instead of just one genome, we have 30 trillion within us.
Oh my
And
gosh.
the numbers are profound. So take an organ like the liver, and the main cells in that organ, hepatocytes. Any two hepatocytes sitting next to each other, next door neighbors, have on average 2,000 distinct changes in their genome. So when you zoom out and look across the entire organ, that means that every single gene in our genome is mutated 2 million times in that one organ inside of our bodies. Now compare that to the world's population, and there's actually more genetic diversity by 5x inside of each of our livers than there is in the world's entire germline. This blew our minds when we saw it, and we began to wonder what this could mean for biology and for medicine.
Oh, that's incredible. So that's an enormous scope. Tell us how you are studying that biology and what are the implications in terms of health and disease?
Yeah, totally. So one of the first things we realized is if there is this genetic diversity inside of our bodies, then there's the opportunity for evolution and natural selection. And it turns out that in the context of disease, certain cells evolve the ability to resist disease due to their somatic mutations. And other cells actually may have mutations that drive disease. And we decided to build a platform that would enable us to interrogate this biology. We call it Sematic Genomics. And it has 4 key steps.
Okay.
The first is we phenotype cells in the organ. We find the cells that appear to have a phenotype, which makes them resistant to disease, or to find the cells that are driving disease. Then the second step is we isolate those cells. So we separate them from the others. Third step is we sequence those cells. And to do that, we use the world's most sensitive DNA sequencing technology. It is roughly 1 billion times more sensitive than the standard sequencing methods that are whirring inside of every next generation sequencing machine in academic centers and in pharma companies on the planet. That can detect a mutation at unbelievable levels. And then lastly, we compute. And we learn from the data we generate, integrate it with other data sources to figure out what changes inside of those cells are driving or preventing disease.
Terrific. So then tell us about how that translates into an opportunity for drug discovery. What does that mean, and how are you creating an engine to do that?
Right. Well, the most important decision that any biopharma company makes, that any biotech makes, is deciding upon the target for a disease. It's the fountainhead for all of drug discovery, for all of drug research. And we have a unique lens upon that. And that lens is somatic genomics. So, we aren't the first people to work in human genetics and want to discover drugs. In fact, human genetics is proven to be the best way to find drug targets. A target supported by human genetics has roughly a 2 to 3x higher probability of success in going through the clinic. And when success rates are only 10%, that's a huge difference maker.
Sure.
It turns out that in 2021, roughly 2/3 of drugs had genetic support. 2/3 of drugs approved by the FDA had genetic support. But we bring a unique lens to this. And there's 3 ways that somatic genomics goes well beyond what's possible with population genetics. The first is the immense scale of what we can do. We only need 10 to 100 patients to discover a target compared to the hundreds of thousands or millions in typical genetics. Second is that the targets that we find are directly selected by the disease biology because we're finding those cells that directly resist disease inside of patients, as opposed to trying to figure out which change in the millions that might be different between you and I in our genome are linked to a phenotype. And the last is the specificity with which we can discover a target. Not only do we figure out what gene is directly mutated to cause a disease or prevent the disease, in some circumstances we figure out the amino acid and the change to that amino acid, telling us exactly how to target that protein or that pathway to cure disease.
Great. So it sounds like incredible scope in terms of mutations, also broad applicability.
Yeah.
Tell us a little bit about where you might be going first with this platform and where you see it potentially growing from there.
Yeah. There's so much breadth of what's possible here that it's been really fun trying to figure out where to start. We've already demonstrated that the technology can find targets in cardiometabolic diseases,
Okay.
in infectious disease, in autoimmune disease, in immune oncology, just to name a few. We're active in neurodegenerative disease and other diseases within those other categories I mentioned, as well as areas like aging. So the breadth is enormous.
Yeah.
And we think about the way that our platform can discover targets with 4 different mechanisms.
Okay.
The first is protecting cells from an extrinsic disease, a disease where patients say have a lifestyle change that leads their cells to be stressed.
Okay.
We can find mutations that protect those cells.
Okay.
We can also find mutations that protect cells from intrinsic diseases, so countering genetic mutations, whether those are a rare genetic disease or cancer or others. We also find mutations that directly cause disease. My favorite example of this is in autoimmunity, where we're discovering mutations that drive immune cells to target self.
Wow.
And the last is finding mutations that enable those same immune cells to target cancer, to fight disease inside of us.
Fantastic. Well, it sounds like you have an incredible platform to get started. I know you've been at this for a number of years, but we're very excited to see what Quotient has in store for 2024 and welcome you back next year as well.
Thank you, Lenny.
Thank you for being here.
It's been fun chatting.
Good.